Hello everyone. In this video, we will embark on a fascinating journey into the world of genetic modification. Our topic today is unlocking the potential of genetic modification. We will dive into the artificial transformation of DNA in organisms. So let's start by defining genetic modification. Genetic modification is the artificial transformation of DNA in an organism using genetic modification techniques in biotechnology so that we can modify the gene expression of that organism. So the history of genetic modifications actually has dated way before technologies arrived. So we have selective breeding, which is our traditional way of husbandry, and also molecular cloning. For animal husbandry, which is one of the ways that we selectively breed animals to improve the desired traits, is also a form of genetic modification. After generations of breeding, these gene expressions of the desired traits will become more pronounced. And also, dog leadership, which is one of the most common example in genetic modification, is one of the product of somatic cells nuclear transfer, which is the transfer of nucleus of a somatic body into an egg that has its nucleus removed. So the resulting cells with the donor nucleus can develop into an organism genetically identical to the donor. And the second thing is embryonic division, which is a process of cell division that occurs during the early stage of embryonic development. This process is fundamental to the formation of an embryo from a fertilized egg, or we call zygote. So several background information before we dive into genetic modification is genome, gene expression, the process of genetic modification, and some vectors that are often used in genetic modification. So what is genome? Genome is an entire set of DNA molecules carries the genetic information that determines the traits of an organism. It is actually a double complementary antiparallel strands linked by hydrogen bond between the pair base, and the base are nitrogenous base, including 4 nucleotides of adenine, cytosine, guanine, and thymine. What is gene expression? So gene expressions involve two processes. The first one is transcription, which is a synthesis of RNA using DNA to produce messenger RNA, or we call it mRNA, that is complementary to the DNA template. And translation is the synthesis of polypeptide using mRNA from that transcription process, creating proteins that will express the desirable traits. So first let's talk about transcriptions, which is the synthesis of RNA using information in DNA. The most important component of this is promoter, which are regions of DNA initiating the process of transcriptions. So these promoters acts as a binding site for RNA polymerase, an enzyme that is responsible for synthesizing RNA. And RNA polymerase will initiate the transcription of the downstream gene when binded to the promoter. The choice of promoter actually influences how tightly or loosely the modified gene is regulated. Strong promoters lead to high expression, while weak promoters lead to lower expression. And look, we also have other key elements such as a core promoter, which contain essential sequences such as a Tata box that provide a binding site for RNA polymerase. And translation is the synthesizing of polypeptides using information in mRNA. This related to genetic modifications in a way that is transferring of one gene from one species to another so that the inserted species develops the same trait with the parent species. So how does it apply to the process of genetic modification? So first we have isolation, which is the process where the genes of interest is isolated from the source organism's DNA sequences. Next, the isolated genes is digested using enzymes, cutting the DNA as specific sequences. For insertion, the genes need vectors such as plasmids, viruses, or some artificial chromosomes to be carried into the target organism. And expression is the last stage of the process when the recombinant DNA will be transcribed and translated in the organism, leading to the production of desired proteins. So, what are the vectors for genetic modification? This will act as medium to introduce a gene of interest into the host organism to produce desired traits. So, the most commonly used are plasmids, which is the DNA molecules in bacterial genetic modification. The components of this include origin of replication, which is a specific DNA sequence where the process of DNA replication begins. It will allow the plasmid to replicate independently of the chromosomal DNA, ensuring that the daughter cells receive a copy of the plasmid during cell division. Next, we have selectable marker, which is a gene that encodes a trait allowing the cells to contain the plasmid to be selected for or against. And we have multiple cloning sites, MCS, which is a region that contains multiple unique restrictions enzyme recognition sites. It will allow the insertions of foreign DNA into the plasmid. Each unique site corresponds to a specific restriction enzyme facilitating the cloning process. And we also have a promoter. In the context of plasmid, a promoter controls the expression of a gene inserted into the plasmid. It determines when and to what extent the gene is transcribed. And lastly, we have the terminator, a DNA sequence that signals the end of transcription. It will ensure that the transcribed gene is properly processed and help regulate gene expression. Also, we have lab experiment in our biology courses that demonstrate genetic modification. And we have bacterial transformation, a process where bacteria takes up a foreign genetic material, often in the form of plasmid. We have transformed cells, which are often called transformants, that will contain the plasmid and the genes of interest that was inserted in multiple cloning sites. We will have selectable markers, which is the genes that help the transformant resistant to antibiotic. We also have plasmid with a gene of the antibiotic act, which acts as a medium inside the bacterial cells to provide antibiotic resistance, allowing the cell to grow in an antibiotic environment. And in Biology 110 course, we study the ampicillin resistant. The plasmid contains the gene for ampicillin resistant, which is AMR gene. The antibiotic ampicillin inhibits cells wall synthesis so that no new bacterial cells can be made. However, when the cell contains a plasmid carrying the AMPR gene, it can be make the enzyme beta-lactamase and break down ampicillin before it affects the cell. So one insight from this is that the extensive and sometimes inappropriate use of antibiotics may create selective pressure, promoting the survival and proliferation of antibiotic-resistant bacterial strains. So how is bacterial transformation used in laboratory experiment? So we have a study of E. coli bacteria that are susceptible to ampicillin because they do not have a plasmid with the RPR gene. Commonly used plasmid in experiment is PUC19, which contain RPR gene and the laxid gene. So we will compare between plates with and without ampicillin of bacterial cells contain or not contain plasmid PUC19. For plate with PUC19 and ampicillin, we can see that there's an insoluble blue pigment which turns the colonies blue. For plate without PUC19 but with ampicillin, we can see no bacterial growth at all. For plate without PUC19 and with ampicillin, or with PUC19 and without ampicillin, we can see that the plate is actually the same because bacteria will grow anyway, even without or with the presence of ampicillin. Alright, we have discussed several background information and lab experiments. So, what are some real world connections of genetic modification? First, we have applications in medical field, agriculture, and environment. For medical field, we can see that insulin, which is often used in diabetes patients, is actually produced in bacteria through the insertion of humans' insulin gene. Similarly, humans' growth hormones can also be synthesized using genetically modified bacteria. For agriculture, we have seen several breakthroughs in science, which has purple tomatoes, which are engineered to have high antioxidants levels so that it can fight disease and promote healthy crops. And we can also see that there are chicken resistant to avian influenza, which can enhance the resistance of poultry to specific disease, contributing to the health of livestock and food safety. For environment, we have several plants that can clear pollutants from the air by collecting nitrogens. One of the example is the American chestnut tree that is modified for enhanced tolerance to environmental stressors. And we also have gene therapy, which is a term used to describe the introductions of healthy genes into cells to replace or correct faulty ones. This holds potential for treating various genetic disorders. For example, in disease caused by a single gene mutation, researchers aim to engineer and deliver corrected genes to restore normal cellular functions. However, we have several ethical concerns. First, about welfare. Genetic modifications raise ethical concerns regarding the welfare of living organisms. In the context of agriculture, genetically modified crops are designed to resist pests or thrive in specific conditions, potentially improving yields and reducing the need for chemical pesticides. However, concern exists about unintended consequences on non-target organisms such as ecological balance and the overall well-being of ecosystems. That also leads to ecological concerns, which is when genetically modified crop is cultivated, there's a risk for gene flow to wild relatives, potentially affecting genetic diversity of native species. Additionally, the use of genetic modified organisms in our culture may alter the dynamics of local ecosystem. And third is the equity in access. Genetic modification technologies, particularly in the context of agriculture and medicine, raise questions about equitable access. There's concerns that certain communities or countries may have limited access to the benefits of genetic modification, potentially exacerbating existing social and economic disparity. So, what are several pros of genetic modification? First, we have pest-resistant crops. Genetic modification allows the introduction of traits that make the crops resistant to pests. This can reduce the need for chemical pesticides, promoting environmentally friendly and sustainable agricultural practices. Farmers may experience increased yields and economic benefits due to reduced crop loss from pest damage. This also have improved gene expression at early stage of development. So genetic modification enables the enhancement of desirable traits in organisms such as improved growth rate, disease resistance, and nutritional content. This can lead to the development of crops with enhanced nutritional values or animals with improved meat quality. So, what other negative impacts? It can have some harm to biodiversity. The introduction of genetic genetically modified organisms into natural ecosystem might pose risks to biodiversity. Crossbreeding or gene flow between genetically modified crops and wild relatives can alter genetic diversity of native species. And we also have several unintended resistance to medicine in bacteria or viruses. The use of genetic modification in agriculture and medicine may contribute to the development of resistance in bacteria or viruses. For example, the overuse of genetically modified crops with built-in resistant mechanism could lead to the evolution of resistant pests. However, in conclusion, GMO is still one of the most powerful scientific tools to save biosphere from climate change and overpopulation if we use it with careful and ethical consideration. So that's the end of my presentation. Thanks for listening.